Well, good afternoon. My name is Lorenzo Pini. I work in the same topic about brain connectivity. In my presentation, I would like to make a journey with you. So the title is From Neurodegeneration to Focal Brain Disease. Why? So in this journey, we will deal with 3 different diseases. Brain tumors, we have already seen the presentation by Alessandro. Then we will move to stroke and degeneration. So basically, we have 3 different diseases. We have glioblastoma, we have seen. It's a rare disease. The median age is 65 years with a lot of variability. And there are some main prognostic factors that Alessandro already showed us. Then we have stroke, that from an epidemiological point of view and from a mechanistic point of view is completely different. We are not speaking about cells that are traveling, but we are speaking about a vascular disease. Also the epidemiology is not a rare disease. We are talking about the second cause of mortality, first cause of disability. And then we have degeneration, like Alzheimer's disease. We are talking about proteins. In Alzheimer's disease, we are talking about accumulation of amyloid, tau, and atrophy that lead to clinical symptoms. Now the question is this, do we have a possibility to find a common substrate to link together these different diseases? And the answer is yes, and I guess that you already guess what I'm talking about. And I'm talking exactly about brain connectivity, functional structure. Basically, it's a feature of the brain that in some way is common across these 3 different diseases. So here I will show some potential models in a very simply way. So when we talk about degenerative disease like Alzheimer's disease, we are talking about proteins, misfolded proteins that accumulate to the brain. And what is interesting is that if we have amyloid and tau in Alzheimer's disease, which are the bad guy of this disease, and accumulate in a specific region, well, the probability to find this, the accumulation, so the longitudinal probability, it's very close to connectivity. So if I have 2 regions that show higher connectivity, and one of these regions, let's say time 0, show accumulation of tau, well, the probability to find longitudinal in the region highly connected is very high. I'm not talking about causation, I'm talking about relationship. So it's still an open question, who is driving who. But the point is that there is a close match between molecular pathology and brain connectivity. And we have seen by the presentation, Alessandro's talk, that the same substrate in some way can tell us some mechanism about glioblastoma. As Alessandro showed that the connectivity between regions, stronger they are, more probably these cells can spread. And then we have brain stroke, that is a completely different disease. There is nothing traveling or spreading. But what happens usually is that I have a lesion, and what I have is that basically I have some local changes. I have a disruption of connectivity, but still there are some effects that some way are spread according to the connectivity. And so there is a kind of functional or connectivity diaschese that has been described by Professor Corbetta 10 years ago. And again, we have, so here, 3 different diseases, 3 different mechanisms, but some common substrate represented by brain connectivity. And I guess you have seen this slide by Alessandro yesterday if he was in the educational course. So basically, if you want to make a very simple example, in the brain there is this kind of system where we have structural railways and the traffic, and the way we can read in the brain this phenotype can tell us something, a lot, about this disease. But what we know about the connectivity? So we have seen that there is a match between pathology, but now we are going to go a little bit deep inside this relationship. And the aim is to provide you with some ideas of what I'm talking about. So let's talk about Alzheimer's disease. We have seen that there is this match between molecular pathology and connectivity. But we also know that this alteration or this match between molecular pathology and brain connectivity follows 2 main axes, which is time and space. So basically there is an interaction of when the alteration of connectivity and the match with molecular pathology occurs, and how, in terms of space, this happens. So when we know from the most extreme study is from Quiroz in Yarumal, is called the Pueblo, the village of Alzheimer's disease. It's the highest concentration in the world with a genetic disease. Well, we know, unfortunately, that in children with the genetic mutation, I'm talking about presenilin 1, this is one of the most known genetic mutation for Alzheimer's disease. Well, already in children, so no amyloid accumulation, no atrophy, no cognitive deficit, no clinical symptoms, but we have alteration in brain connectivity, at least this study suggested 10 years ago. And what is interesting is that time influences space, because what happened at the beginning is that these networks doesn't show disruption, so hypoconnectivity, but show a kind of hyperconnectivity. This is an open question whether this hyperconnectivity is compensatory or it's an imbalance between networks. So we know that in Alzheimer's disease there are some networks. So the deformal network, which is probably some of you is familiar with this network, showing the disruption of connectivity. And we have the competitive one, which is called silence network, showing hyperconnectivity. So basically, if in theory, if I have a disease that affected this network in terms of hyperconnectivity, and also in terms of molecular match, I should observe hyperconnectivity in the other. And that's exactly what we know from frontotemporal dementia. So it's another disease. This is linked with accumulation of tau. There is no amyloid. The network which show the earlier vulnerability is the silence one, and what we know is that the competitive one show hyperconnectivity. So basically, what we have now, what we know, are two important concepts or features, is when this alteration occurs, and also the interaction between time and space. And in this perspective, it's a little bit provocative, but it's linked with what Alessandro was referring. I mean, now we could link this information, or at least we should start discussing whether we could include this information, for instance, in the development of drugs pipeline. We know that in Alzheimer's disease, unfortunately, we have the Food and Drug Administration these years approved 3 drugs for Alzheimer's disease, anti-amyloid, but the European Medical Agency rejected the approbation. So there is still something that we have to understand, and probably brain connectivity could help to fill the gap. At different level, we can use as a marker to assess the efficacy of the drugs in some interim analysis. Or since we know the time, we could use as a enrichment strategy, because we know that basically assessing the alteration of brain connectivity, we know that there is this mismatch between hyper and hypo, or we can use as target selection. And I will give you an example of target selection because it's the most tricky one. Alessandro was talking about brain stimulation in glioblastoma. So in the way we have done brain stimulation in the past, I want to improve a specific function, let's say executive function or memory, and what I do is the targeting of anatomical specific regions, try to modulate anatomically some anatomical region. But now we have enriched the information of our brain, so what we can do is targeting a networks, a disease-specific networks. And this could probably maximize the clinical benefits. It's something we have tried. I won't go into details, but this is just to show you that what we have done in Alzheimer's disease and frontotemporal dementia, which is the two diseases that we have seen showing the opposite pattern. Basically, when we target these networks, the effects is different whether we are targeting the hypo-connected network or the hyper-connected network. When we apply stimulation to the network showing disruption, hypoconnectivity, our patients show an improvement in cognition. When we try to target the network showing hyperconnectivity, our patients show improvement in clinical symptoms, but not in cognition. This effect was also matched in terms of connectivity. So basically, we have some information about how we can target, how we can modulate this network in our patients, trying to maximize the clinical benefits, and so also techniques and some information to drive the therapeutic cognitive effects or the clinical effects. This was a pilot study. The sample size was very small, but that was just to show the feasibility of this kind of approaches. Of course, it's not a slam dunk yet. There is a lot of things that we have to understand, but now we have this information that I was referring before. We have the time, we have the space, and so we can personalize in some way, try to create a tailored intervention in this kind of patients. And what is interesting is that, as we have seen, these the space, there is a hyper and hypo that we have shown. I show you in Alzheimer's and frontotemporal. We can observe also in Parkinson, in Huntington, and in stroke. Stroke, which is not a degenerative disease. In this graph, there is no glioblastoma, because, as Alessandro was pointing before, the connectivity studies in glioblastoma are quite new, so we are understanding the connectivity fingerprint of glioblastoma. And before I neglect this panel, the main prognostic factors, as you can see, there is none of the guidelines or prognostic factors take into account the host, the brain, as Alessandro pointed before. And as he has shown, there is an interaction between glioblastoma and connectivity. And again, we can use this information. I will skip this part since Alessandro already presented, but this point is that what we have seen in Alzheimer's disease about using this network information, trying to developing a new therapeutic strategy can be translated to the glioblastoma field. And finally, we have stroke. Stroke, we know there is a local disruption, a neural death of the ischemic core. We also know from animal studies that there is locally, we have some alteration that can be reversible. But we also know that these alterations are widespread and seems to follow, as we have seen before, the connectivity of the brain. So basically, in this slide, I would like to point out that when we have a lesion, we have some first level of disconnectivity, so a first level of alteration, but as we have seen, there is also a second level of disconnectivity. So the alteration seems to follow this connectivity pathway. It's something that, these are some preliminary results, just to give you an idea what I'm talking about. So if I have a lesion in a specific region, when in animal, let's say the connectivity match of alteration in stroke has been assessed using in animal studies, in which we can control experimentally the lesion and to assess the contralateral changes. This is something that we can do in humans, of course. But what we can do is to assess whether the lesion contralaterally is connected or not. So we have two different patients. These patients, this is the lesion, which is affecting these tracts, and the contralateral region is, there are some tracts which are connecting or affected in this case, while in these patients, the two regions are not connected. So I won't expect alteration in these contralateral regions compared to this. And this is exactly what we have observed. The change in the microstructural integrity of the contralateral regions is highly related to the degree of connectivity of the lesion. So basically, if the contralateral region is highly connected with the lesion, is going to observe higher degenerative effects. And what we also observed in this study, this preliminary study in stroke, is that there is a degeneration over time of the connectivity pathways. And this effect is widespread, so the full connectome show these degenerative effects. This is a linear mixed model, but you can look at these columns, so the p-value. So this means that the connectome show degenerative changes both in the ipsilateral hemisphere, which makes sense, is where we have the lesion, but also in the contralateral. And also the tracts that that are connecting the lesion show this degenerative effect. But what is interesting is that whether acutely there is a relationship between the connectivity alteration with behavior, this effect is decoupled longitudinally. So basically what we observed is we demonstrate this two level of this connectivity, so this first level, and the second level, which is not linked with the recovery of these patients. And so basically, in terms of intervention, this make an important point, whether we should target or not the structural connectivity, since this is not linked with the behavioral recovery. So this is another example of how we can use this information, hopefully, to drive this kind of intervention. Now, I talked to you about these three diseases. So we have seen how degenerative, glioblastoma, and stroke show this close match between connectivity alteration and the pathology. And I show you these three different diseases in relation in connectivity because it's something that is close to the BigBrain's project that Professor Corbetta was mentioning. So just eBrains is this, I know most of you know eBrains, this is a European digital infrastructure in which there are several open science tools. We have atlases, we have seen by Timo presentations where in eBrains we can link different scales with different functionality and modalities. We have seen also inside eBrains there is modeling and simulation where we can create these digital twins of patients. And inside the eBrains 2.0 project, the WP2, we are enriching the platform with data in the framework of what I just discussed. So with data from degenerative and focal disease in terms of glioblastoma and stroke. These are the partners of the WP2 packages. So we have in red, we have the clinical centers which are providing the clinical data of patients with degenerative stroke and gliomas. We have the blue partners which are providing the pentamodal dataset. So we are enriching the platform with 200 healthy controls, highly characterized in terms of macro scale. So we are collecting structural, diffusion, functional, metabolism, and high density AG. And this is also integrated with the yellow centers, which are the intracranial AG for epilepsy patients. So the framework that I just showed you, the idea is to integrate inside this project under this framework of connectivity. So in the first task, we are going to build a platform for the integration of these different data and to manage the data and also to process the data in order to extract these Connectome features. And task 2.2 and 2.3 is the pentamodel acquisition. So these 200 healthy subjects characterized by this pentamodel acquisition, structural, diffusion, function, metabolism that I already mentioned. And then the task 2.4 is what I was referring, the inclusion, the harmonization of this clinical data set to build the Connectome features that can be useful to answer to several research questions. We are including data from brain tumors, brain stroke. For brain degeneration, the use case is represented for the moment by Parkinson's disease. And is enriched also with intracranial AG from epilepsy patients by our partners. And we have also the idea to create a kind of harmonized protocol in order to provide the European communities, the European clinical communities with a common acquisition protocol, both in terms of MRI, in terms of clinical. And the aim is we will propose, within our consortium, we will have this proposal, and inside the European Academy of Neurology, this proposal will be discussed and will be approved and then shared among partners in order to harmonize some way the acquisition of this data, and this will provide a way also to merge the data all together. So instead of using thousands of patients, hundreds of patients, maybe we could use in the future thousands of patients harmonized. And thanks for your attention. These are some persons in the lab of Professor Maurizio Corbetta that are working on this topic. And thank you for your attention.